Hi everyone, my name is Angelica Gomes Ueltschy. I am a research scientist and the director of the Mesoderm team at Trailhead Biosystems. And here at Trailhead, we use our proprietary technology, the high-dimensional design of experiments, to differentiate pluripotent stem cells to specialized cell types. The HD-DoE technology allows us to check multiple factors at the same time, multiple signaling inputs. And after we do that, we identify the cell phenotype by doing different and several measurements of gene expression. These allow us to create a mathematical understanding of how genes are regulated and allow us to generate novel protocols for specialized cell types. On this poster today, we are going to be focusing on the production of hematopoietic progenitor cells out of induced pluripotent stem cells. This differentiation protocol is a multi-stage protocol composed of 3 stages. The first stage is patterning the cells to the mesoderm lineage. After that, we use a stage 2 media to make hemogenic endothelial cells. And finally, the stage 3 media guide our cells to the hematopoietic progenitor cell state. On this protocol, we did several harvests at different time points because the hematopoietic progenitor cell state is an unstable state and changes quickly. So you have early, middle, and late harvest. Additionally, we created 2 different recipes for the stage 3 differentiation. The first recipe was based on proliferation of the cells, and this recipe contains the usually used cytokines that you see in the literature. The recipe 2 was a recipe that was generated to optimize the expression of HSC genes identified by Calvanese on Nature 2022. And this recipe will therefore be expected to maintain better the hematopoietic progenitor cell state. So here you have flow analysis of the different harvest days of the 2 different recipes that were generated on stage 3. So here you have early, middle, late harvest. Here we can see high CD34 expression and CD43 expression as well, showing that the cells are on the track of making blood. Also, you have high expression of CD90, which shows how early the cells are. On the middle harvest, you have also high expression of 34 and 43, and a little bit less expression of CD90. It's still high, but lower. In all the 3 different harvest days, CD38 and CD45RA was low expressed. Finally, on the late harvest, we still have CD34 and CD43 close to 80 or 90% of expression, however, lower CD90 expression. After doing flow cytometry analysis of the cells, we cryopreserve them and we check the post-thaw viability by flow cytometry. Here you can see the data from cytox staining, and now our cells from the different time points, they show high post-thaw viability, close to 80%. In order to understand the phenotype of the cell, we did gene expression analysis, and we measured key genes related to the hematopoietic stem cell phenotypes, such as HLF, MECOM, SPINK2, MLT3, RUNX1, and CD45. And here you can see that CD45 expression increases over time. You can also see that some genes, the expression changes over time quickly, such as HLF and SPINK2, and other genes are more stable, such as MECOM and so forth. So to better understand the phenotype of the cells, we did bulk RNAseq to identify what are the cell types that we have in our culture. So here you have bulk RNA-seq analysis of different samples. All the way on the left, on the dimensionality reduction plot, you have stage 0 cells in light blue. In the center, we have our hemogenic endothelial cells, the stage 2 cells. And on the right, you have cells from different harvest time points and from different recipes. Here, you can see the progression of the cells of the recipe 1. You have early cells here on green, and then you have middle cells in red, and then late cells in orange. So the cells are going from here, from the top of the graph, moving all the way down to the bottom of the graph, showing that these cells are progressing fast over time. Now I want you to pay attention on the recipe 2. The recipe 2 was the recipe that we focus on upregulating the expression of the hematopoietic stem cell genes. This recipe, you can see the early and late time points in pink and orange, and these cells are clustering together, the early time points and the later, showing that the cells on this recipe, they progress in a slower speed, indicating us that this recipe can maintain the stem cell phenotype over time better than the recipe 1. When you compare here the early recipe 1, the green is up here, and the late recipe 1 is down here. So after identifying the transcriptomic profile of the cells, we did functional analysis of the cells. We did colony-forming unit assays from the 2 recipes and also from early, middle, and late harvest. As you can see, in earlier harvest, we have the BFU and CFU-E colonies in higher number, and in the later harvest, you have more of the myeloid colonies in higher number. The potential of the cells change over time, as you can see, and as is supported by the phenotype of the cells that you saw on the dimensionality reduction plot, also changing over time. After doing the colony-forming unit assay, we decided to measure the differentiation potential of the cells using direct differentiation protocols to erythroid lineage, neutrophil lineage, monocytes, and some for the lymphoid lineage. As you can see here, you have the early, middle, and late harvest being differentiated to the erythroid lineage. Here you have CD71 and CD235A expression. And as you can see, all the harvest days and all the different recipes have the red blood cell potential, as you can see by double expression of CD71 and CD235A. Here, for the neutrophil lineage, we have CD11B being analyzed and also CD66B. Additionally, we check CD14 expression to rule out the presence of monocytes in our culture. And as you can see, the cells, the hematopoietic progenitor cells designed by Trailhead have neutrophil potential, as you can see by the CD11B and CD66B expression. Here on the bottom, you have the monocyte potential, which is high in our cells in both recipe 1 and the recipe 2 in the early and late harvest. We have expression of CD33 and CD14 expression. And we have high expression of both markers in the different recipes and time points. Finally, for the lymphoid potential, we use commercially available kits, and then here you have the presence of CD3 positive cells being shown in different harvests of cells and different recipes. In the recipe 2 and the earlier time point gave the better lymphoid potential, showing 62% expression of CD3. So that's all. Thank you very much for your time, and thank you for the interest in the poster. I am open to answer any questions in case you have. Have a good day.